I don't remember exactly what you said about a flu shot. Did you say, just tell me over? Was it not to take a live virus or what?
No, the flu shot is safe. There are two flu shots. There's a regular dose and there's a high dose. And from the point of view of CLL, I think the more of the vaccine you get, the better. So take the high octane flu vaccine. It may or may not do you any good. One of the things that we're discovering actually in multiple myeloma is that that patients with multiple myeloma, which is a bone marrow tumor, they don't make very good antibodies against vaccination either. But Revlimid or lenalidomide has been used to treat those patients, and they do much better with vaccination. So as we go along, we may well be using lenalidomide to enhance the response to a variety of vaccines.
With this triple shot, the flu vaccine, do we still get a booster in January or February?
I think it depends a little bit on what happens during the flu season. Bill?
So even in people who have normal immune function, I don't think most people realize this, but the protection that you get is only about 2 to 3 months. So if you get the vaccine and you're real high risk in, say, October or November, it's not a bad idea to get boosted in January, February, and that should...
two will be a triple whatever?
Mm-hmm.
Okay, thank you.
One of the slides, I believe it was Dr. Plunkett, showed the proliferation or impacted group of CLL cells in the marrow. And I got the impression that because they're where they are and clumped as they are, they're protected somewhat from the treatments that are given. Aside from bone marrow transplants, is there any other consideration being given to treating the bone marrow of CLL patients directly rather than orally or through infusion?
A strategy that's being taken is to use a small molecule that will pry the CLL cells away from the protective cells in the bone marrow. So this is presently in our clinic, and the theory is that that the CLL cells then will get into the peripheral blood, as Michael and I said, that they may die naturally there, or they will be more susceptible to chemotherapy. So that's a strategy that's investigational. But you might want to say something about this same compound in mobilizing stem cells.
What do you want me to say? So the drug is called plerixafor, and it has been approved by the FDA as a molecule to enhance collection of stem cells because it facilitates stem cells leaving the bone marrow and circulating in the blood. So the trial that we have open now is plerixafor with rituximab, and the plerixafor is given to disrupt this interaction between the stroma and the CLL cells so that the CLL cells will go out and circulate into the blood, and that's when the rituximab can attack the CLL cells.
The other thing is, the vegetable drug, the PEITC, that in actual fact, the stromal cells are keeping the CLL cells alive in a number of different ways. But one of the ways is that they can provide an amino acid that the CLL cells can't make themselves. So they're feeding this small molecule. And one of the things that happens with the PEITC is that it interferes with this, what they call the glutathione pathway. Now, Peng has actually developed a derivative from this, which is again a very small molecule that's sort of like a fake amino acid. And instead of the stromal cells pumping this good stuff in, it gets converted into pumping in a poisonous substance that destroys the ability of these particular cells in this particular model system to stay alive. So that's the closest that we have got. One of the problems is that the stromal cells in the lymph glands and the bone marrow, etc., are very similar to stromal cells in every tissue in the body. So that if we were to attack the stromal cells, we would attack widespread, so that it would go away from the selectivity that Bill has mentioned. But I think that some of the work that Peng has been doing is really going to shake things up a lot. So that one of the things that we tend to forget, Dr. Freireich, who has taught all of us here, always used to say that leukemia was the window into cancer. If we could understand what was happening with leukemia, we could understand cancer. And I think that we're finding this reverberation, that we find that some things that work in one leukemia, we can develop that concept and apply it to another leukemia. Some things in solid tumors that we can apply to all of the leukemias, etc. So one of the great things about working in a big cancer center is that we keep on getting fed by our colleagues with additional information, and it's a nice thing.
If I may.
You may.
I, just as an addition to your question, which is relevant and that I didn't specifically mention, is that the strategies to reduce these pro-survival proteins act independent of this bone marrow protection and stroma. So we're thinking that taking care of two problems at once with these as we go. You may want to say something about it, the activities in the clinic, Bill, of SNS or the transcription.
SNS.
Tumor lysis or biological activity.
So we did a trial with SNS-032, which is a drug that's like flavoperidol, which is now being studied, and they're doing work to gain FDA approval for flavoperidol. The drug that we worked with is called SNS-032. It works similar to flavoperidol. There's another drug that Merck is developing that works similarly, and clearly there was activity that we saw with SNS-032 in the clinic in patients who were on that study, and there's work that's being done with Merck, with the Merck compound, that shows activity with that compound as well.
And you know, one of the things that Bill's lab has shown is that if you give flavoperidol or SNS-032 or this Merck compound, which was originally developed by Schering, is that you knock down these survival proteins. But one of the ways that the cells recover is that they make more of the survival proteins. And Bill and his lab have found a couple of molecules that are very effective in stopping the leukemic cells from recovering, because it stops them from making these new copies of these molecules. So we think it's going to be a new advance on something that we know that works, and I think we'll be able to make it more effective and certainly more user-friendly than we had in the past.
You mentioned Revlimid several times tonight. Is that actively being used as a treatment, or is it still a trial type program? And is it given to you via infusion? And my last question related to that would be, when would you use Revlimid over the FCR that you talk so glowingly about?
So Revlimid is approved by the FDA. It's approved for two diseases, multiple myeloma and a specific subgroup of patients who have myelodysplastic syndrome. It's a pill, and so it's approved by the FDA, but it's approved for other diseases. Work that we've done, a significant amount of work that we've done, has shown there is activity with Revlimid or lenalidomide. In the treatment of CLL, it's active by itself. It's more active when you combine it with things, particularly antibodies like rituximab or ofatumumab. So it's not approved by the FDA specifically for the treatment of CLL. They're working on clinical trials right now that will demonstrate activity in CLL, and hopefully it will be approved by the FDA for treatment of CLL. Clearly, we've demonstrated in our studies that there's activity against CLL. The activity is a little bit different than chemotherapy. So patients have to stay on the drug for many, many months before we see their best response. We see responses after a few months on treatment, but the best response doesn't occur for several months after patients have been treated on the drug. And generally, for the patients that are on our studies that have had good responses, at least the patients that I have, Dr. Keating can comment on his, they've stayed on the drug. So I've seen, in the last couple weeks, I've seen patients who have been on lenalidomide for more than 3 years. They've had a nice response, they're in complete remission, but they're staying on the drug. So it may be approved by the FDA for CLL. It's not yet approved. It's not yet in the compendium, for example, for the NCCN. So the National Cancer Treatment Network has what's called a compendium, where they go through different drugs and go through the data for those drugs and say, well, these drugs are active in these diseases, therefore Medicare should pay for them in these situations. So it hasn't made it into any compendium at this point. So it's generally not covered by insurance companies or by Medicare because of that. It's not paid for, it's very expensive, and so it's not generally used in the treatment for patients with CLL. It's not as active as FCR is, particularly in the frontline setting. So FCR is much more active, much more effective at getting patients into complete remission, which is what you want. It's better tolerated by patients over 70 years old. So that's the patient population that we've sort of targeted and have been working on developing treatments that are better tolerated, and lenalidomide or Revlimid is one of the drugs that we've studied in that patient group.
So one of the things that you have to appreciate is that it's very important for us to get an activity signal from a drug, so that there's enough enthusiasm for us to go to the next level, the next level, the next level. You know, one of the things that that I think that you have to realize is that along with survival proteins and death proteins, there are survival signals for interest in a drug, and death signals for interest in a drug. And the investigators get tired of doing experiments that don't work. So if you actually get something that says, this is doing something important, we don't understand it yet, we just have to get a signal that encourages us to keep on going. Because, you know, being a scientist and being a doctor, it's actually hard work. You know, you get disappointments, etc. Working in cancer centers, there are always disappointments, surprises, etc. So we have to get this little fillip all the time just to go along. And, you know, it's amazing how you have one patient in the clinic that has an unusual response, and it goes like a buzz around the place and says, oh, such and such is working. And, you know, the buzz happens and everyone gets very excited about it. And then if it's a drug that Bill's working on that we may or may not understand much of, as soon as it actually gets to have some activity, we go to Bill and say, can you explain to us what actually is going on? And he does a pretty good job of that.
Sometimes it works the other way around, where we learn how the thing works and then say, this will fit your pathophysiology, man.
The real heroes of clinical research are the patients that allow us to do unproven things to them because there's no other option. So I personally, on behalf of Bill and Bill, would like to really thank you all for giving us your time. We are your agents, but you're our agents. So thank you very much for coming along. And we'll try and do something like this again next year at the International Workshop on CLL. Thank you very much.